Hi, my name is Maddie Cramer, and with my partners, Eve and Rahul, we would like to present to you Operation Growth, Dendritic Rescue, an experimental approach into the morphology behind the cognitive symptoms of schizophrenia.
Our experiment is on schizophrenia. This is a neurological disease that affects 1.1% of adults in the US every year, making it quite common. It is a chronic mental disease that affects a person's cognitive and behavioral traits. Some cognitive symptoms that we'll be focusing on are working memory and learning. These take place in the prefrontal and medial temporal lobes. The major problem with schizophrenia is there's no cure, and relapse is quite common. So we'll be exploring some ways where we can mitigate these cognitive symptoms. So our proposed solution to kind of ease the cognitive effects that schizophrenic patients suffer from would be to assess its relationship to pruning of dendritic spines. As we can see on the graph on the right over here, the red schizophrenic patient has a much less density of these spines than a healthy patient in the blue. And to do this, we would focus on targeting and overexpressing specific genes using CRISPR-Cas9 prenatally in rats to stimulate the growth of these dendrites, mitigating the effects that come with memory and learning. So here's a little more on the gene overexpression via CRISPR-Cas9. So as mentioned before, this would be done prenatally in the rat model. And CRISPR-Cas9 is a highly specific gene editing tool that has been proven time and time over again to be effective to either insert or delete a certain gene sequence. And so in this experiment, we'd be overexpressing the target genes that are related to dendritic growth, and delivery would be achieved prenatally via injection into the amniotic fluid. The three genes of interest that we'd be focusing on to overexpress via CRISPR-Cas9 would first be MAP2, which is important in determining the shape of dendrites in development. Next would be CRTC1, which has links to memory and learning, having an important role in synaptic plasticity. Lastly, NRG1, which has been shown that a reduction of NRG1 leads to inhibition of NMDA receptors, which means less glutamate in the synapse, thereby affecting memory and recall. Since we are looking at both the changes in dendritic growth as well as effects on cognitive symptoms of schizophrenia, we'll have two different hypotheses. The first one is if the MAM model rats, which are schizophrenic rats, have their expression of MAP2, CRTC1, and NRG1 prenatally stimulated using CRISPR-Cas9, they would demonstrate a higher density of dendritic spine growth in the Golgi stain of the prefrontal and medial temporal lobe. This will be shown on the left over here. We have an example of what the overexpression will look like, and on the right, that's what we'd expect for the growth to kind of look like in the dendrite spines. The second hypothesis relates to enhancing memory and learning, where we will be focusing instead on how the cognitive symptoms of schizophrenia present in rats that have their target genes overexpressed via CRISPR-Cas9. We'd expect these schizophrenic rats who have the overexpression of these genes to show a decrease in cognitive symptoms related to memory and learning. We would quantify these symptoms using the Morris-Water maze and the novel object recognition paradigm. So hopefully we'd see that they would have better performance if they had overexpression of the target genes compared to the control that would not have the overexpression.
Our methodology had two experimental groups and two control groups. The manipulations for the experimental groups were performed prenatally. Each group was then subjected to a Golgi stain assay to observe dendritic spine growth, as well as two cognitive assessments. These assessments were the Morris-Water maze to test for spatial learning differences between groups, and the novel object recognition test to test for changes in memory. The variables, groups, controls, and methods are outlined in detail on subsequent slides. The graphic on the right illustrates the various groups used in the study. Neither of the control groups were subjected to gene editing via CRISPR-Cas9, but one control group expressed the MAM model for schizophrenia. This offers a basis of comparison for both variables. In contrast, the experimental groups both underwent gene editing, while the MAM model inducement was only performed for one. Variables that were controlled for throughout the experiment included living conditions, food, and water provisions, which were all under ad libitum. In addition, the method of MAM model administration and genetic manipulation was consistently performed prenatally, while the cognitive assessments were all done postnatally. Here we can see an illustrated experimental design timeline. At first, the MAM model is induced prenatally, as well as a CRISPR-Cas9 system. Then, the Golgi stain assay as well as the 2 cognitive assessments are done postnatally. The experimental design used groups of 5 rats for each control and experimental group. For the groups that underwent gene editing, 3 groups were used for each gene that was edited. These genes were MAP2, CRTC1, and NRG1. Each rat was then subjected to a Golgi stain assay to observe differences in dendritic morphology. The rats underwent 3 trials each of the Morris-Water maze test and the novel object recognition test to test for spatial learning and behavioral memory.
We will now look at our results, first in terms of dendrite morphology. Groups with schizophrenia but without gene editing did have a significantly different number of spines from the control. However, groups with schizophrenia with gene editing, specifically the NRG1 and MAP2 genes, were not significant in terms of number of dendritic spines from the control. We will now look at the second part of our results, which was the cognitive assessment. As a reminder, the cognitive assessment was done using the Morris-Water maze and novel object recognition, and it was done 1 year postnatally. It was found that the schizophrenic group with gene editing of the NRG1 and MAP2 genes did not have significantly different scores from the novel object recognition and Morris-Water maze paradigms compared to the control group. It is important to understand that the interpretation of these results is that the NRG1 and MAP2 gene editing produced cognitive assessment scores that were not significantly different from that of a normal rat. This interpretation supports the idea that increasing dendritic spine growth by editing the NRG1 and MAP2 genes helps reduce the cognitive effects of schizophrenia by increasing this learning and memory ability. As a reminder, the end goal of this experiment was to help improve the cognitive functions of individuals affected with schizophrenia, so we're really excited by these results, and we hope that they could progress to have potential treatment in humans. Looking at our figures, we will first look at figure 1.a. This figure is specifically for the gene editing with the MAP2 gene, and it's in terms of dendrite morphology. The key piece of this figure is highlighted in the red outline box. In this box, we have a comparison between our control and our schizophrenic group with MAP2 gene editing. The schizophrenic group with MAP2 gene editing is not significantly different in terms of dendrite spines from the control. However, we did observe that the group of schizophrenia and no MAP2 gene editing was significantly different in that it had significantly less spines from the control. Figure 1.b is very similar to figure 1.a, except for this figure we are focused on gene editing with the CRCT1 gene. However, with the CRCT1 gene, we observed that there was a significant difference between our schizophrenic gene-edited group and our control. This can be better phrased as the schizophrenic group with gene editing had significantly less spines than the control, which was not observed with the NRG1 and MAP2 gene editing. Figure 1.c follows the same trend as figure 1.a. As a reminder, this figure is specific to the NRG1 gene. It is observed that within our highlighted red box, the MAM, aka the schizophrenic group, did not have a significantly different number of spines from our control group. This is something we want to highlight in our results, that the MAP2 gene and the NRG1 gene did not significantly differ from the control, but the CRCT1 gene did. Here we have our 2 figures for the cognitive assessment using the Morris-Water maze. Once again, we've highlighted our main results in the red boxes. These red boxes indicate where the error bars do not overlap, which is indicative of significant differences. Figure 2.a is in reference to the MAP2 gene editing only, and the significant differences you see in the red box are due to the schizophrenic group without gene editing and the rest of the groups. This means that the schizophrenic group with gene editing was not significantly different from our control. The same lack of a significant difference between the schizophrenic gene-edited group and the control was also observed in figure 2.b, but this was specific to the NRG1 gene editing. So our final figures are for the cognitive assessment specific to the novel object recognition task. Once again, the red boxes are indicative of where the error bars do not overlap, which is also indicative of significant differences. So figure 3.A is specific to MAP2 gene editing. The significant differences that we observed were between the schizophrenic group without gene editing and the rest of the groups. This means that the schizophrenic group with MAP2 gene editing was not significantly different from our control. Figure 3.B shows these same results once again, but this is specific to the NRG1 gene editing, but once again, the schizophrenic group with gene editing was not significantly different from the control. This returns to the idea that these two cognitive assessments, they had scores that were sufficient with schizophrenic groups and gene editing because these scores were not different from that of a normal rat without schizophrenia.
The ROSE study has several drawbacks in clinical and practical applications. Primarily, the MAA model proves to be difficult when observing altered lobe functionality as a result of schizophrenic effects. These functional differences need to be inferred from behavioral assays, as direct observations require the use of complex models. Further, the CRISPR-Cas9 system of gene editing often introduces mutations easily as a result of nonspecific targeting. In addition, long-term effects of psychosis that mirror humans cannot be observed in rodents as they have shorter lifespans.
As any other neuroscience experiment, there's always room for improvement. These are some general ideas on future direction or follow-up to this experiment. First, we could determine some other appropriate genes that we could overexpress via CRISPR-Cas9 to promote dendritic growth. Next, we could attempt the CRISPR gene editing postnatally rather than prenatally in this experiment to compare fetal versus adult environmental factors and how that plays into a role of schizophrenic symptoms. Lastly, we could see if NRG1 and MAP2 show significant differences in the dendritic growth with different cognitive symptoms. So this would be assessing the improvements in performance with attention and social cognition, for example.
This study offers significant contributions to research in preventative neurobiology by linking behavioral and morphological differences associated with schizophrenia to specific genes. Further research can be done to combat the onset of this disease by targeting MAP2, CRTC1, and NRG1 specifically. In addition, this study further solidifies the notion that dendritic degradation is rooted in effects of schizophrenia, offering a niche for research regarding dendritic growth promoters to combat the behavioral symptoms of schizophrenia.